we are really looking forward to listen to Professor Sugiyama to give this talk. Just a little bit about the background of Professor Sugiyama. Currently, he serves as the head of the Sugiyama Laboratory in RIKEN Innovation Center, and he's also the professor emeritus in the Graduate School of Pharmacology in the University of Tokyo. And he served as a consultant in many drug companies and research institutes. And he's the chairman of many organizations as well, including the ISSX, International Society for the Study of Biotics and metabolism and disposition, and the board for the Pharmaceutical Sciences of FIP, which is the International Pharmaceutical Federation. Okay, Professor Yama,
okay.
For your presentation.
Thank you very much for your kind introduction. First of all, I want to express our great thanks to Javi and also Director Peter for inviting me to this University of Auckland, ABI, Bioengineering Institute. So today I want to share with you our recent development on this title, the targeting of small molecules to liver by transporter analysis using PBPK modeling with statins and anti-HCV drugs as examples. So let me talk to you. Direction is totally opposite. Okay, thank you. Is it working?
Yeah.
Okay. So this slide shows the current programs in drug discovery and development from now on. This shows the program. This slide shows that this one means clinical phase 1. Clinical phase 1 study is done by using the healthy volunteer, basically. So then go to the clinical phase 2, 3, then regulatory, the examination by regulatory authorities, and also finally approved. So assuming that the 100 compounds go into the clinical phase 1, after having done so many preclinical studies using animal models or so many in vitro experiments, about 40% of the compound drop out to go into before going into the phase 2, and then that is this kind of clinical data. This is taken from 2004 paper, but even now it is almost the same. This attrition rate is very high. So this kind of information indicates that the low productivity, but that is almost 90% of attrition rate and high cost, $2 billion US per marketed drug of drug development. And much of attrition takes place in phase 2 and beyond. So why so? That is a very, very basic question. So this slide shows the factors governing the inter-individual variability of drugs effect and side effect. Actually, if we can develop the drugs for, genuine rat, SD rat, it is very, very easy because we don't need to worry about the inter-individual variability. But actually, to develop the human drug, it's not so easy. So after the drug is given at certain dose or certain dosage regimen to human, that reaches the blood circulating blood. Then after that, it goes into the target tissues. So then when it gets into the target tissue, that may exert some some pharmacological effect and also side effect. That is then in this pharmacokinetic phase, the many enzymes, metabolizing enzymes and transporters, and also in the pharmacodynamic process, the molecular targets such as receptor or enzyme for endogenous compound play a role. So therefore, to understand the drug action or drug side effect, we have to consider the inter-individual variability of enzyme transporters and also the receptors or molecular target. So the compound with drug-like properties should have the minimum inter-individual variability and minimum drug-drug interaction and optimum ADME properties. ADME is absorption, distribution, metabolism, and excretions. So why do we have such a high attrition rate after getting into clinical trials, even though pharma industries have done so many preclinical studies, animal model or in vitro model? Of course, nobody knows the exact reason, but I guess those two major reasons may account for this phenomenon. One is a poor predictability from animal experiment to human. We already know great species difference. Second is a poor predictability of the exposure in the target tissues for pharmacological and toxicological effect because of the active transporters in the uptake and efflux process. So that means there is a great inter-individual variability, great interspecies variability in the ability of transporters. So even if this drug goes into the rat brain very well, it does not get into the human brain so much. We know many of those examples. So how to overcome those problems, but before that, let me show you this data. This is a difficulty, this slide shows a difficulty to predict human bioavailability from animal studies. Bioavailability is a fraction of the compound which reaches to the circulating blood after oral administration. Okay, that is a bioavailability, definition of bioavailability. So this y-axis shows the human bioavailability and x-axis shows the animal bioavailability. So this is the dog, rodent, and primate, monkey, something. So if you look at this data, there are such a great interspecies difference. No animals can predict the human bioavailability. So the further analysis indicate that these species difference mainly due to the difference in fast pass metabolism in the liver and also small intestine, and also fast pass uptake by the transporter in the liver and the intestine. So that means the passive permeability does not show so much species difference. That is well known. So therefore, such a species difference is explained by the species difference in metabolizing enzyme and also the transporters. So we have to know those species difference. Recently, while I was in the University of Tokyo and also in RIKEN, we did the government-based project. It is a NEDO research project. Title of the project is the Establishment of Evolutionary Drug Development with the Use of Microdosing Clinical Trials based on the quantitative prediction technology of ADME. So this this grant was provided by NIDO, that is the New Energy and Industry Technology Development Organizations. I'm a project leader. So then in this project, we did perform 27 clinical studies. So for the 4 years. So then in average, in 1 year, we did about 7 clinical studies have been performed. The idea of this project is by using the microdose. Microdose is 1% of the dose compared with the therapeutic dose. So that is a very small dose. So if we administer the drug at microdose level, then we can measure the pharmacokinetics of those compounds, and sometimes we do measure the tissue distribution by using PET imaging, PET probe, positron emission tomography probe. So by doing so, even in human, we can measure the tissue distribution of the compound. So by integrating all of those microdose data, including the tissue distribution, we can finally predict the pharmacokinetics and also tissue distribution of the compound from the microdose to the therapeutic dose level by using the kind of mathematical model, including the physiologically based pharmacokinetic model. That kind of idea. So then, I'm very happy that the NIDO gave us quite a lot of research grants for 4 years. So then we did lots of in vitro and also clinical studies. So then many of the outcomes shown today from those NIDO projects. So then before going further, let me show you the drug transporters. As you may probably know, some of you may probably know, the transporters can be divided into 2 categories. One is a SLC type of transporters, solid like carrier, and the other is a ABC transporters. That is a transporter having ATP binding cassette region in the cytoplasmic side of the membranes. So then, it's known that the SLC transporter mainly function as an uptake transporter into the cells. But the ABC transporter mainly function as an efflux transporter from the cell to the outer environment. I have been involved in the transporter studies for the last almost 30 years, since the first transporter was cloned in the world. But actually, while we are getting, not only my group, but also many other groups, whenever we found very new data, it struck me very much because it really makes sense from the rational localization of the transporter. For example, the localization of the major ABC transporters, such as P-glycoprotein or MRP2 or BCRP. Those kind of major ABC transporters are located in the liver and kidney on those apical membrane. That makes sense, because in the liver and kidney, excretion of xenobiotics is really necessary. So then, the important ABC transporters are located on this membrane, which can excrete the xenobiotics from the cell into the urine or bile. That really makes sense. But on the other hand, the same ABC transporters in the blood-brain barrier or in the intestine is located on the apical membrane again. But here in this case, those ABC transporters protect the entry of xenobiotics from the blood circulation into the brain, or from the intestinal lumen to the blood. So in that sense, ABC transporters for such the brain or intestine function as a barrier for the entry of xenobiotics into the important place, that is sanctuary. So then, it's about the already 13 years ago, when the transporter chapter came out into the well-known textbook of pharmacology, Goodman-Gilman. So now we have the 13th edition. Last year it was published from the publishers. So this kind of figures just came out in those textbooks. So now we know that the transporter play important role in the tissue exposure of drugs leading to pharmacological and adverse effects. So then I always say to the people in the pharmaceutical industry, necessity for changing from ERA to control plasma exposure to ERA to control tissue exposure. Transporters are major determinant for governing the tissue exposure of drugs in terms of drug-drug interaction and pharmacogenomics. That causes the inter-individual variability. Because as you may easily imagine, for the last 50 years or even more than that, the pharmaceutical industry have been measuring the plasma exposure because it is easily sampled, even in clinical studies. So therefore, but in many times we know that the plasma exposure measurement are really not enough, because target for pharmacological effects and the side effects are located inside the tissues. So we somehow have to predict the exposure in the tissues. Let me show you one good example. This is a transporter related to intrahepatic circulation of non-metabolized statins, such as pravastatin or rosuvastatin or pitavastatin. Okay, so statins are drugs to lower the blood cholesterol. Okay, so many people are taking statins, including me. So when you get the statins orally, it is absorbed through the intestinal wall. Again, we need a transporter, uptake transporter, OATP2B1. Then from the hepatic portal vein, it is taken up into the liver cell by OATP1B1 and 1B3. That is our very old paper. We first demonstrated the importance of those transporters, then followed by the biliary excretion by MRP2 and or BCRP. So therefore, for pharmacokinetics of statins, at least 3 different transporters plays a very important role for its enterohepatic circulation. Intestinal absorption transporters, hepatic uptake transporters, and also the biliary excretion transporters. Because of the presence of those 3 transporters, this statin is always exposed in the liver. That is a very good nature, because pharmacological effect of this drug, that pharmacological target of this drug is HMG-CoA reductase enzyme. So therefore, this drug is always exposed to the liver. That is a good nature. And also, particularly, the very efficient hepatic uptake by OATP1B for statin exists in the first pass hepatic uptake of statin. That also shows gives a good nature, because most of the drug is taken up into the liver cell by first pass. So therefore, fraction which is escaped into the hepatic vein, followed by the liver circulation, can be minimized. So therefore, for those reasons, I always say, those transporters give this drug a good nature, high pharmacological activities and low side effect. So actually, when I made this kind of presentation, probably around 1997, that is already 20 years ago, many pharmaceutical industry people are very much surprised because by that time, the transporters are well known to be a bad actor. Bad actor. That's because the P-glycoprotein MDR1 had been first cloned and well known. But P-glycoprotein always function to decrease the entry of drug into the brain. So therefore, because of the P-glycoprotein, the CNS drug is not so active at all. And furthermore, P-glycoprotein also exists in the intestine. So after the oral administration, because of that, the efflux activities, orally drug is not available into the circulation. So therefore, because of the P-glycoprotein initial discovery, many people believe that the transporters are really bad actors for drug discovery and development. But actually, it's not necessarily the case. These transporters play good actors. However, this is a very disaster. Disaster take place about the starting again. So that is the 2001. So therefore, more than 15 years ago, when the new statin, cellular statin, was developed by Bayer company in Germany, it had a very good reputation because the cellular statin is very, very active for lowering the LDL level. Actually, it is true. But very soon after it went into the market, 52 patients in the world died. And among 31 patients who died in the US, 12 were given also GEM500, another lipid-lowering effect. So certainly this kind of disaster is at least partly explained by the drug interaction, other drugs. When this news came out in the world, my group had already started analyzing this mechanism. So to make a long story short, I just want to show you only the final conclusion. The GEM500, that's the co-administered drug, lipid-lowering drug, that is metabolized to glucuronide in the liver cell. And that glucuronide inhibits the major metabolizing enzyme of cellular statin, CYP2C8, almost irreversibly. So therefore, the CYP2C8 content in the liver is decreased very much. In addition, GEM500 is efflux out into the circulating blood, then it inhibits OATP1B mediated hepatic uptake of cellular statin. So in that sense, the GEM500 functions as a dual inhibitor for uptake transporters and also metabolizing enzyme. So because of this nature, with the co-administration of GEM500, cellular statin concentration in the circulating blood is increased very much. So that may increase this drug concentration in the muscle compartment. So if the concentration is increased in muscle, that may cause the rhabdomyolysis. The rhabdomyolysis, the muscle is lysed. So it's a very, very severe symptom. So then some people really die. So since this news came out, as I just mentioned, my group quantitatively analyzed this drug-drug interaction. So then we published several papers showing that not only metabolizing enzyme, but also the transporter play a very important role in the drug-drug interactions. So then US FDA paid much attention to this transporter-mediated DDI, then decided to form the ITC, International Transporter Consortium, that consists of regulatory agency and also several pharmaceutical industry and academia people like me. So initially, ITC started from around 10 members. So nowadays it is almost 15 members, but we are still very, very active. The very important point is that we have a good collaboration with regulatory authorities and also pharmaceutical industries. So this is a little bit different story, but as some of you may know, I got this description from the latest cited from WebMD. So actually, the hepatitis C, most of the liver cancer comes from hepatitis infections, particularly hepatitis C. So until recent time, the HCV is a very, very dangerous disease. And then, but most recently, for the last, I would say, 6-7 years, the pharmaceutical industry discovered a very nice drug to decrease the hepatitis C virus. That is very, very effective. So nowadays, the hepatitis C is not so dangerous compound because it is cured by hepatitis C drug. So then, as you see, most of the time the new medicine remove all traces of the virus from your blood within 12 weeks, with some as soon as 8 weeks, such as these compounds. This is called virus clearance, and it's what doctors look for to determine if you are cured. How long you need treatment can vary. It may range from 8 to 24 weeks or more. Anyhow, it is a curable drug just by the simple 1 or 2 drug. And then you don't need to worry about side effects so much. But as you see here, those hepatitis C drugs are taken up into the liver very much. That makes sense because the molecular target exists in the liver. So then, those hepatitis C drugs are the substrate of hepatic uptake transporters and also hepatic efflux transporters, as well as many CYP enzymes. So for both, so far, as I told you, for both statins and anti-HCV drugs, molecular entities which are recognized by molecular target, that is HMG CoA reductase in case of statin, and the hepatitis C virus in case of the HCV drug. So then, the recognized by molecular target and the hepatic uptake transporters, or ATP1Bs, seems to be similar. That is the reason why many companies developing statins and anti-HCV drugs make OATP substrate even after the lead optimization process. So actually, in the drug development, company first try to find out the lead compound. And after that, they do lead optimization so that its pharmacological effect could be maximized and also side effect can be minimized. Even after those kind of lead optimization process, almost all the recently developed HCV drug or statins OATP substrate. That means the structure of molecular target and also OATP might be at least locally very similar. So now I want to move to the pharmacogenomics of OATP 1B1. That is a model-based analysis. I just want to tell you very simply this one. My group published a paper almost 15 years ago showing the clear clinical evidence saying, indicating that the genetic polymorphism of OATP, that is the hepatic uptake transporters, that mutation caused the change in the pharmacokinetics of pravastatin. So that was also supported by our in vitro experiment using cyto-directed mutagenesis type of study. Since this data came out from our group, many people did lots of similar studies using probably more than 30 compounds so far published. So the OATP is now well-known polymorphic transporters, which may cause the inter-individual variability of many compounds. So actually, this is one of the papers published by other group, published in New England Journal of Medicine, one of the best clinical journal. They did the so-called GBAS. GBAS is a genome-wide association studies. So therefore, they did not target any gene. They searched the genome on the wide range, genome-wide association studies. And then they try to find out the responsible gene responsible for the simvastatin-induced myopathy. So then their analysis indicates that this OATP 1B1 polymorphism is a very important, most important the gene. That is, if you compare the wild type and mutant type homozygote, the odds ratio is almost 17-fold. So this is a very important polymorphism. That makes sense for us when this data came out, because with this polymorphism, my group already indicated that plasma exposure is increased very much. So therefore, the muscle exposure will be increased very much. That may cause the increasing the myopathy, the probability. So then after that, there are many other clinical type of study, many other clinical type of the big scale clinical studies. Rosuvastatin case, the N number is more than 3,000 patients. Using those 3,000 patients, they analyze both OATP1B1 and also BCRP. BCRP is one of the efflux transporters. They show that the pharmacological effect, LDL cholesterol decrease level, is associated with the mutation of BCRP. On the other hand, OATP1B1 polymorphism is not associated with the pharmacological effect, but that, though not shown here, but that is associated with the myopathy. That is a side effect. That is consistent with the previous finding by other group. So let me summarize many of those reports so far published by many other groups. Basically OATP1B1 polymorphism has no effect or small effect, if any, on pharmacological effect, HMG reductase in the liver. On the other hand, that OATP1B1 polymorphism has a big effect on adverse effect, particularly myopathy. So that is a summary of many publications. How can we explain this result? But let me summarize so far. All of these pharmacogenomics and DDI studies on OATP1B1 suggested that the hepatic uptake plays an important role in the plasma clearance of therapeutically important drugs. Why did this polymorphism and/or DDI affect only side effect and not pharmacological effect? And then to understand that, the PBPK modeling should answer this question. Then we published many PBPK modeling studies. That is the first study done in 2009, published in 2009, using pravastatin as a model compound. This is an intravenous administration pravastatin. This is a oral administration pravastatin in human, into healthy volunteer. Actually, we did analyze this result by using this PBPK modeling. And the basic parameters shown here, hepatic uptake and also biliary excretion and also some small metabolism or basolateral efflux. So in those parameters are essentially obtained from in vitro experiment and then in comparison with animal experiment, both in vivo and in vitro. So then we did the extrapolation into human. This is not fitting result. This is an extrapolation, prediction result from basically in vitro to in vivo. So both after oral administration and IV administration, we can describe this plasma concentration profile very well. So based upon thus established the PBPK modeling, we did the sensitivity analysis. For example, if we change the hepatic uptake function, this is a normal state, the threefold and one-third. If we change the hepatic uptake abilities, we can, the exposure in the plasma, exposure of drug in the plasma is changed very much. But on the other hand, even if we change the hepatic uptake ability, exposure in the liver is not so much changing. That is really consistent with the previously obtained clinical data. Because the clinical data said that the polymorphism of OATP1B1 affect the side effect, myopathy, okay? But no effect on the pharmacological effect. The target is a liver in that case, but here in this case, target is a muscle, therefore plasma exposure is very important. On the other hand, if you find the BCRP or MRP2 polymorphism, that is the efflux transporters, even if you change that activity over a wide range, there is no change in the plasma exposure, but there is a big change in the liver exposure. That's because the final output process is changed, so therefore liver concentration is very much affected. The reason why plasma concentration is not so much affected is explained by the hepatic uptake limited clearance. But today I don't go into the detail, but that can be easily explained by much more simpler modeling. But this sensitivity analysis indicates that the change in the hepatic uptake ability alters the plasma concentration profile sensitivity, toxicity therefore, and may not affect the profile in the liver target tissue. GWAS for simvastatin, in fact, demonstrate it was the case. Alteration in the biliary excretion ability by MRPs and BCRP may affect the pharmacological effect much more sensitively than that of the uptake. This prediction is consistent with the most recent studies published by other groups, such as Jupiter trial or simvastatin, the GWAS studies. So now I want to tell you the challenge of regulatory authorities, that is US, and this is EA, EU, and also this is Japan. DDI guidance, use of the PBPK modeling. Now, everywhere in the world, the regulatory authorities are encouraging the use of PBPK modeling for the prediction. I don't go into the detail, but I already explained to you the International Transporter Consortium is formed. So then they published the white paper first in 2010 in Nature Review and Drug Discovery. So based upon this kind of white paper, they recently issued the guideline for predicting drug-drug interaction. So actually, this is taken from the review article of FDA scientists. Okay, so FDA really, let's say, recommend to use the PBPK modeling in predicting drug-drug interaction or effect of polymorphism or extrapolation from adult to baby, and then the prediction of the changing pharmacokinetics under the organ dysfunction condition, like the liver dysfunction or kidney dysfunction. So then actually this is a example of the use of PBPK modeling in the application of drug approval process. Actually, this was taken from the paper published in 2013, but the last year the similar data was published again, but it does not make so much difference. Actually, in many cases, this PBPK modeling is used for predicting DDI, followed by pediatrics prediction or something as you see here. So then, this is a very good example. Use of PBPK modeling is the application of drug approval process. Actually, that's the most important point. The many pharmaceutical industry are using PBPK modeling because of the example. In the official labeling, the labeling, all the drugs, when you use, the user use that. The user may read those labeling. So a user or a doctor will read that labeling. In the official labeling, the prediction of DDI using PBPK modeling has been appearing for the last several years. We already have many examples. For example, the new drug, ibrutinib, without doing any, they did some clinical studies, but the other data is obtained by only the simulation. But even if we use the simulation, if the regulatory authority find that is a reasonable simulation based on the reasonable modeling, that can be described in the labeling without any clinical evidence. That is really surprising. I can never think of that possibility 20 years ago at all. And then that also came to the transporter substrate. That is one of the HCV drugs, the cimepravir, which the Janssen discovered and also developed. Then it's already appeared in the market. So then, for this drug, that is a very good OATP substrate. So even for this kind of drug, without doing all the clinical studies, they showed the characteristics of this compound, particularly DDI, in the labeling. So now finally, I want to, using just 10 minutes, I want to show you the use of PET image to confirm whether the PBPK modeling based on the in vitro measurement of transporter can predict the drug exposure in the liver. Because in the previous slide, I already showed you the sensitivity analysis or something like that. But that is not based on the PET imaging. That is just based on the plasma concentration profile and also urinary excretion profile. We did not measure the hepatic uptake directly. We did not measure the biliary excretion directly. So then, to be honest, the modeling result shows that is the case, but we are not so sure whether our modeling is really correct or not. So therefore, we decided to do the, let's say, PET imaging. So then, by using the PET imaging, we can measure not only the plasma, but also the target tissue like the liver or kidney or brain, or even the not only liver but also the biliary excretion, etc. So therefore, that is the advantage of the use of the PET imaging. So as you see, this is the principle of PET imaging, but we usually use carbon-11 compound or O15 compound or F18 compound. Usually half-life is not so long, for carbon-11, 20 minutes. Even F18, the most frequently used the PET imaging using F18 is the fluorodeoxyglucose method. Fluorodeoxyglucose method, that PET imaging can measure the glucose consumption in all the tissues. So that is used for the diagnosis of the cancer in some of the tissues or some other functional tests. So therefore, what I want to tell you is that because of the relatively short half-life of these molecules, then you have to synthesize the molecules near the bedside. So soon after it is biosynthesis, it is synthesized, we do the very quick test using animals. Then we go into the clinical study. Of course, we have to get the approval of IRB in the hospital or the major researchers. So then, in collaboration with Dr. Watanabe in RIKEN Kobe, in my group, we have been synthesizing the many PET probes for the transporters in the brain or kidney and the liver and the intestine, etc. So then, using those probe compounds, we have been doing the PET imaging studies. And then our most recent paper came out in the DMD, drug metabolism and disposition, because the picture color is very beautiful. It appeared in the cover page of this. Our paper came out in the cover page. So then, let me show you the actual data. But my group, they synthesized the dihydropravastatin. That is a little bit different from clinically used the pravastatin. The structure, just a minor change. But my group did the experiment using in vitro system. This is a hepatic uptake, OATP1B1 mediated uptake process, and this is saturation kinetics to determine the Kn values for 1B1 and 1B3. And then comparing those characteristics between dihydropravastatin and pravastatin, it is very similar. Basically no change in the characteristic. That's why we use the dihydropravastatin for our PET study. This is actual PET imaging. This is a whole body. The 15, 35, 55, something, some minutes. Initially, that goes into the liver, very high. So then, followed by, this is a, later I can explain to you, but gallbladder or bile duct, it goes in. The later time, it goes into the urine. But then, if you look at this, the lower panel, this is a control experiment in human, and this is a rifampicin-treated co-administration rifampicin. Rifampicin is well known to inhibit OATP1P, uptake transporter, and at the same time, biliary excretion transporter, MRP2. The data just came out as we expected, if you look at this, the image. Finally, then if we show you the time course of this data, we do blood sampling after the administration PET image, PET probe. This is a blood sampling in the control and blood refurbishing co-administered human. So this is a liver. The concentration in the liver, in the control, and in the presence of refurbishing inhibitor, the uptake is very much limited. Then this is a biliary excretion. This is a control, and also this is a refurbishing co-administration. This is a kidney. Kidney does not show so much difference. Actually, those really make sense because refurbishing inhibits the hepatic uptake process and also the biliary excretion process. Then we do the mathematical model-based analysis. But today, I just want to show you the very simple analysis. In my laboratory, it's called integration proto-analysis because the uptake takes place from the blood compartment into the liver. So if we measure the time course of the drug in the liver as a function of blood concentration time profile, just using this simple differential equations, if you integrate both left-hand side and right-hand side, you can come up to this kind of simple equation. So therefore, ordinate is a liver concentration, and the x-axis is a AUC in the blood divided by the concentration of drug at time t. So then you can get the straight line for the initial stage. So then that process, that slope will give you uptake clearance. What the theory can tell us. Actually, the result just came out as we expected. The same kind of integration plot is obtained by also for biliary excretion. The y-axis is the biliary excreted amount, x-axis is the AUC in the liver. So then from this slope, we can calculate the biliary excretion clearance. Actually, by doing those kind of analysis, we obtain the hepatic uptake clearance in control 11 and refurbishing co-administered decreased to half, and also biliary excretion clearance also decreased to 60%. So then we of course did the more complicated mathematical modeling to analyze the PET imaging data, but basically the obtained data is almost the same as we obtained in this type of similar, very simple analysis. So now, my idea is shown here. By using the PET image, we can get the uptake clearance and also biliary excretion clearance, or even the base lateral efflux clearance. So far, including my group, we are obtaining those kind of uptake and biliary clearance just by the mathematical modeling based on the plasma profile and urinary excretion. That's not so reliable. So therefore, after we repeat this kind of study, clinical studies, using several probe drugs, we can compare that's obtained in vivo uptake and biliary excretion clearance with in vitro, the measurement. We already established many good methods for in vitro. So then, from this in vitro data, we can make a prediction, but the PET imaging can give us the in vivo data. So therefore, we can make this cycle, then we can finally obtain the good scaling factor from in vitro to in vivo for uptake process and also the biliary excretion process. That's what we are doing in my laboratory. However, that costs very much. So then, and also we need lots of manpower. So that's why the consortium research is now being held by inviting 6 global pharmaceutical industry. So that is a pre-competitive collaboration, 3 hours collaboration. So then 3 years collaboration. This is the final year. So then we really have to finish up all the data analysis. But anyhow, the PBPK-based prediction using loss obtained scaling factors can be compared with that obtained PET imaging data. For example, the increase in drug exposure in the liver by inhibiting the biliary excretion, etc. That is a very important point, because this is the only reliable in vivo information obtained by PET imaging. Plasma profile is really not enough. So that is the take-home message in my whole presentation. It's essential to use a scaling factor for the particle uptake and biliary excretion clearance between the in vitro and in vivo to make the IVIVE successful. Because that is a very important point I forgot to tell you. Of course, if the company can make a PET probe when they develop the new drug, it's very nice. However, that's not so easy, particularly for intermediate scale of the company. It is almost impossible. So therefore, once we can establish this kind of method and we can obtain the good scaling factors, the company can use the only in vitro data, then use this scaling factor to predict the in vivo phenomena. Because from the preclinical data, we can easily say that this compound is taken up into the liver cell 80% by 1B OATP1B1, 20% by OATP1B3. And the biliary excretion takes place 70% by BCRP and 30% by MRP2. Those kind of information can be easily obtained by the in vitro experiment. So therefore, once we know each scaling factor, we can predict those data. But of course, we have to verify whether that prediction is good or not. So we are making the database of drug-dependent PBPK parameters: uptake, metabolism, biliary excretion, urinary excretion, and inhibition and induction, and fraction of enzyme responsible for that metabolism, or fraction transporter responsible for that transporter, for as many drugs. These in vivo parameters will be obtained by optimization method using clinical data. We then can predict any kind of DDIs between the multiple drugs. That is my dream, but actually it may take for another 5 years to come to this kind of thing. Finally, I'm a COI, Science Advisory Board member of the following companies: SimSip. SimSip is the biggest simulation company, published, launching, everybody in my area knows SimSip. Actually, that is not only the software, but also lots of database about the space, ethnic difference or effect of organ dysfunction or something, the many informations with software ability. So then, actually, you may be surprised that when the company have the one license from this SimSip, they have to pay $300,000 US per license and per year. But nevertheless, for example, Pfizer has the 10 licenses. But of course, the cost is not proportional to the number of licenses. I don't know exactly, but maybe Pfizer is paying more than $1 million US, I'm sure. So then actually even the Japanese company, the largest 7 companies have that license. But that depends upon how they think. If they can avoid unnecessary clinical studies, it easily costs $1 million US to do one clinical study. So therefore, now regulatory authorities are in favor of this kind of model-based simulation. So that is not such a big money for the companies. So I'm having a consultant when collaborating with 30 domestic and global pharmaceutical companies, and then this is a member of my laboratory. So actually, even now, I retired from the University of Tokyo, and my associate professor took over my position. So therefore, I'm still collaborating with my former student or collaborators very well. So then, that is the end of my talk. Thank you very much for your attention.
Thank you for very insightful, very wonderful presentation. Question from the audience. Yeah, Brian.
Well, thank you for a very interesting talk, and I'm going to demonstrate my lack of familiarity with the field. But the slide that you put up at near the beginning was quite alarming, showing the lack of correlation and bioavailability between animals and humans, even for primates.
Yeah.
So, is it understood to what extent that reflects changes in specificity of the transporters or in expression of transporters and metabolizing enzymes for that matter? Or do you think both are equally important? Is it expression or?
Actually, today I don't have enough time to show you all the analysis, but the picking up the compound of which how can I say, first pass GI or first pass hepatic metabolism or excretion is not done by, let's say, CYP3A4 or some of the enzymes, who are mainly mediated by passive diffusion. There is no such a species difference. So therefore, it may certainly indicate that this kind of species difference is explained by the CYP3A4 and also several other enzymes and some of the transporters. So, but about the details of the contribution of each transporter or each enzyme, it's not well done. I mean, not well analyzed.
Well, to another way to asking that question is, in the in vitro studies, are there major differences in the equivalent homologs across species in specificity?
In for example, transporter
Substrate specificity.
Substrate specificity. Talking about substrate specificity, that's a very good question. Not only my group, but also many other groups did, let's say, using typical 20 substrate, okay? Using typical 20 substrate. They measure the rank order, of the activities for human transporters and rat transporters and monkey transporters. Rank order is almost the same.
Okay.
There are some exceptions. However, the absolute value of the activity is very much different. Mainly the V maximum value.
Okay.
That has been already well analyzed.
Okay.
Peter.
So the thing that alarmed me a little bit in that bioavailability chart was, quite apart from the species difference, was just the variability within a species. And I'm just wondering, when you do your comparisons between the PK predictions and the clearance from the PET imaging, how much are you using patient-specific information to adjust the parameters of the predictive model to compare against the clearance, the PET clearance?
Yeah, in this case, in future, I really would like to do what you said. But at this moment, we are using the healthy volunteer, not a patient. Right. And then, when we select the healthy volunteer, the age is not so much different. Then we are using only male, for example, this study, and then the renal function is normal, or hepatic function is normal. So therefore, the
variability is less.
inter-individual variability is not so much.
But do you still try and use some, I mean, presumably you're using some sort of guess of tissue mass for the different tissues that you're interested in?
That's true. Of course, for each individual,
Yeah.
we measure the tissue mass,
Right.
liver mass. So therefore, we do include those information into our model.
Yeah.
But that effect is here in this case not so much.
Right.
But talking about the patient, that's really very much, I'm sure.
Yeah.
That's what we have to do in future.
Question from Stephen?
I guess any comments I have around the high variability that you get in the expression of drug metabolizing enzymes and also drug transporters in humans, and you're not going to see that in the same degree in animals. Do the PBPK models take that fully into account, and is it able to, I guess, predict how an individual patient is going to respond based on, not knowing maybe what the expression of these particular enzymes or transporters might be in that patient?
Yeah, of course, what you said is very important. We will take that into account. But now, now for the last 10 years, the science has been developed so well, then people already succeeded in measuring the protein expression amount, either transporter or enzyme or isoform, or even the molecular target by using LC-MS method.
Yep.
So then nowadays, at least in healthy volunteer and we already know all the transporter, almost all the transporters or enzyme expression level in the liver, kidney, and also intestine, and to some extent in the blood-brain barrier. So therefore, using those average value, once we could get the in vitro transport activities using expression system of each transporter, then we can measure the expression level of transporters in the expression system. By using the mathematical model, we can extrapolate from in vitro to in vivo. But that is of course, theory can tell us we should compare in vitro extrapolation to in vivo, but nobody really, in the true sense, compared those extrapolation from to in vitro in vivo. That again, my laboratory is now extensively working on that project.
Okay, so let's thank again for Professor Sugiyama for your wonderful presentation.
Thank you.
And thank you.
Thank you very much.
And,
Thank you for joining
we can
us.
have a